Good morning, everyone. Thank you all for joining us today to celebrate World Parkinson's Day together. This is our first celebration of World Parkinson's Day at the Dementia Research Institute, and we hope it's the first one of many celebrations together, because this day belongs to you. We'll be joining the Light Up Blue for Parkinson campaign by PD UK, as you see, by giving up a little bit of touch of blue to our lecture theatre. And also we are wearing blue colors, that as you may have noticed, is both the colors of our Dementia Research Institute and PD UK. In our agenda for the day, we'll start in a few minutes with a few short research talks, but by some of our early career researchers. We'll have a comfort break in between, so around 12:30. We'll have 10 minutes to stretch and stand a little bit, use the restroom, etc. And towards the end, we'll join the campaign Points for Parkinson by playing a poem for one of the participants in the Points for Parkinson campaign. And at the end, when we move to the little room again, we'll be participating in the Sit to Stand Challenge for World Parkinson's Awareness by Neuroheroes. And now, without further ado, I leave the stand to our Deputy Director, Professor Philip Taylor, that will introduce you to our DRI Institute.
So first, it's my pleasure to welcome you here, both in person and virtually, to the Dementia Research Institute at Cardiff. The Dementia Research Institute at Cardiff is one of 7 centres that was recently set up roughly 5 years ago, primarily by the UK government MRC, to energise research into dementia within the UK. These 7 centres, each with their own specialties, are designed to function as a single institute. And the vision of the institute is to energise dementia research in the UK by bringing together the best biomedical and clinical research in the UK with the aim of improving the care of people with Parkinson's and other dementias. Now, the way we're achieving this is with many multidisciplinary teams, so teams with different expertise working together to understand what causes the diseases and in what ways we can help. So this is a brief look at the UK Dementia Research Institute at Cardiff. There are a number of principal investigators leading these multidisciplinary teams, and they can be broadly divided up by the disease that they are primarily studying. The main emphasis at Cardiff is using the information we get from you guys, from your genes, what that tells us about Parkinson's disease and how we can treat it. In red here, it shows you the principal investigators that work on Alzheimer's disease. In green, it's Huntington's disease, and what we're most interested in today is Parkinson's disease, our principal investigators shown in blue. So we have here, for example, Kaya Weber and Cynthia Sandor. These two are both computational biologists, so they use computers to understand the disease processes by studying both your genes, the clinical data they get from you, and also from experimental data. Also, we have Dane McKelly here, who his primary role is in studying the brain cells experimentally, understanding what's different about them, what's changed, and what could be causing the disease itself. And lastly, Meng Li here works with a very special kind of cell called a stem cell, and she can take these cells from humans and grow them in a dish and make brain cells in a dish. So we can actually study human brain cells in a dish. Now, you'll be pleased to know that I'm going to get out of the way and hand you over to the experts that work on Parkinson's disease in a minute. But just a brief introduction to them. The way our research works here is it's really focused right from the start, from the patient, from the person with Parkinson's disease who's living with it, and from all the clinical and genetic data that you guys kindly give to us to enable us to conduct this research. And as you'll see, this is used in many different ways. For example, it can be done via a computer, and Nabila Rahman will be the first person to talk here today, immediately after me. She takes the clinical data and she performs all sorts of mathematical analysis in computer to understand what might be going on. And then the next three talks that will follow will be more experimental. And they use what we call models. So systems that we can study, which in some way tell us something about genes and disease in humans. And this will be through the use of flies, believe it or not, through the use of mice, and then through the use of those special stem cells I told you about that make brain cells in the disc. So, without any further delay, I'm going to hand you over to Nabila, who will introduce you to her work on the computational study of Parkinson's disease in blood. Thank you.
So, hello everybody. I'm Nabila Rahman. I work at the Sander Lab at the Cardiff University, and I'm a researcher here. And what I primarily work on is blood cells, where I basically look at blood cells in your blood to try and advance personalized medicine in Parkinson's. So as Phil said, I am an informatician. So basically I sit behind a PC analyzing lots and lots of data. And this data is basically all the clinical examinations, the blood tests, genetic data, questionnaires, and so on. And I analyze all of them to try and figure out what makes you unique as a person with Parkinson's. So, as you know, the Parkinson's is a complex condition in that each one of you manifests Parkinson's in a different way. Take, for example, Susan here. She developed Parkinson's, was diagnosed with Parkinson's at the age of 55. And now at the age of 60, she has mild shakes and some trouble sleeping. Tom here, on the other hand, was diagnosed at the age of 42. He too now is 60 years old, and he's exhibiting moderate shakes and no trouble sleeping. When you give both these people a drug called X, you'll find that Susan here responds really well, but Tom doesn't. On the other hand, if you give them drug Y, this time Susan doesn't respond well, but Tom responds really well. So my research is trying to figure out something in your body, find something in your body that will tell the doctor whether you're somebody like Susan, who will respond well to drug X, or somebody like Tom, who will respond well to drug Y, so that he can then go on to prescribe you the right medicine. So how do we tell whether you like Susan or whether you like Tom? Well, there are a number of tests we can do. We can take some fluid from your spine in something called lumbar puncture, or we could do a DAT scan, which is taking an image of your brain, a specific part of your brain. And then differences there will tell the doctor what medicine you may need. But the problem with these tests is that they require special equipment and specialist equipment and training, which makes it difficult to provide it on a large scale and open it up to healthy volunteers. And as you know, healthy volunteers are very important for research. And that's why I focus on bloods. Now, you may be asking, why bloods? Parkinson's is a brain disorder, but evidence shows that signs of Parkinson's is actually also reflected in the blood. So if we can look at a simple blood sample, which is cheap and easy to extract, the doctor can tell what medicine you need, hopefully, without you having to go through further testing, such as lumbar puncture or DAT scan. So in that hope, I'm doing research to find some difference in your blood that will set you apart from Susan, from Tom, or somebody who doesn't have Parkinson's. So how do I go about finding this difference? So what I do is look at differences in cell composition inside your blood. By cell composition, what I mean is, in your blood sample, there will be many different types of cells. Some will carry around oxygen and nutrients, and some will protect you from germs by gobbling up germs as soon as they enter your body. And there's a delicate and fine balance of how much of these cells are present in your blood. And if you have too many of these cells, sometimes they will be doing their job, gobbling up cells, for example, germs, for example, coming into your body, but they can also cause unwanted side effects. And in fact, research shows that some people with Parkinson's do have too many or too little of some types of cells compared to healthy counterparts. So I say some because not all people with Parkinson's have this difference in cell types. For example, Susan here, the number of green cells in her body, in her blood, for example, is the same as the number of green cells in the blood of Steve, and he doesn't have Parkinson. Whereas Tom here, on the other hand, has too many of these green cells. So while you wouldn't give Susan any therapy based on these excess green cells, you'd give Tom and people like him who has too many of these green cells floating about in his blood some kind of therapy that would prevent any side effects from these two excess green cells, or even prevent the making of these green cells, which you would hope would give you some kind of health benefit, whether it's decreasing the symptoms of Parkinson's or even hopefully improve prognosis in the future. So to summarize, what my research hopes to do is take a simple blood sample from you, count the number of cell types, for example, these green cells, and then ask, do you have too many of these green cells? If not, you would go for a different kind of therapy. But if you do have too many of these green cells in your blood, we would hopefully give you some kind of drug that would give you some kind of health benefit. Of course, this is still research in progress, and a lot of work needs to go into making sure we are categorizing you into the right group. And as I mentioned, all of this happens on the computer. I simulate and predict what's happening inside your blood. So of course, it needs to be validated by other researchers, like my colleagues here who will be speaking here today, who will validate the results in flies, mice, or even human stem cells to see if it's actually doing what I'm predicting. So that is the gist of my presentation. Feel free to ask me questions. I'll also be there to chat with you later on.
For the DAT scan and other scans you take and the blood cell checks, are you closer to identifying what causes Parkinson's in the first place, and some people rather than others, or is this purely a treatment-based approach?
This is more okay. The question, let me just repeat it for the virtual audience. So the question is, are we close? What I'm looking at, whether it's closer to whether it's more about finding the cause of Parkinson's or diagnosis. The answer to that is this is more about a diagnosis and stratifying you into subgroups to make sure you're getting the right treatment rather than the cause in this case.
Thank you.
Really, if you want to know what's causing Parkinson's, you've got to go really right back to the beginning. You've got to ask why. Why do some of you have more green cells and yellow cells and so on? How? What caused it? When? Was it somewhere in my youth, or was I born with that? Is it a genetic factor? Did I have a traumatic incident years ago which caused it? And we seem to be missing all those answers at the moment. Answers to why, how, when, where, what caused it. I think it's good that you're moving on to the next stages, but we still need somebody to go back and answer those questions.
I'll just repeat what you said. We need to move on and see how and why these green cells, for example, would be higher in Parkinson's compared to healthy controls, and try to figure out what is causing it with what event in your life. In that regard, I am looking at prodromal individuals as well who have not developed Parkinson's and are sort of predicted to do so in the future. And we're also looking at whether they're having a higher or lower level of specific cell types. So if we can sort of map at what point this is happening, we could hopefully be closer to an answer on whether this is a causal effect of Parkinson's.
Sorry, could I ask another question? You're looking at blood cells, and you mentioned lumbar puncture and that scan. Is blood cell correlation, or is it nearly as good as the other ones?
Is lumbar puncture results as good as blood cells? Currently, lumbar puncture is a bit more definitive. A lot of work needs to go into blood cells to make sure more research is needed to sort of extract the data better. Also, we look at it at the same time, like the CSF samples and the blood and see how they correlate. And once we are able to properly make sure we are getting the results and the data as clean as possible, I think we can get something just as well with as CSF.
The reason I asked the question, I'm taking part in a drug trial in Italy, and they wanted me to have a lumbar puncture, which I flatly said no. And they worked very hard to get me to answer them, which I wouldn't. They seemed very disappointed in that, as if it was the crown jewel of basis of research.
Right. In regards to whether CSF is better than blood, currently CSF is more definitive because blood is still experimental. So the clinicians won't be able to do much with blood yet. So this is what I'm trying to find out if something is definitely there that can be used. So you would currently, lumbar puncture would be recommended.
Yeah.
Yes.
There's some questions virtually.
Yes.
Colin Young. Says it's a really interesting presentation. Is there any evidence that L-DOPA, as we use it as a treatment, actually affects cell immunity, given that this is the main treatment?
Should I repeat it?
Sure. Yeah.
The question is if L-DOPA is affecting the blood cells. So far, I haven't looked at the people on drugs yet. Currently, I'm just looking at people off drugs. So that's something, definitely a question I'd have to look at, address in the future. So I cannot answer that yet.
Yeah, there's one other question from Krishna Kumar, but it was very similar to our question earlier, which was to look at whether or not your data is going to tell us about the treatment side of things or the potential initiator and cause of it. I suppose it's a bit of colonnade, needs the information, as you were saying.
Okay, is my data going to answer the causal or more of a treatment side of things? It all depends. If we find these cell types are like going up in prodromal individuals, it might be more of a causal answer. At the moment, I don't know, because I'm hoping it's going to be causal, but more analysis needed. I need to do more statistics and more analyzing to make sure it's causal. It's a question I'm working on right now.
Dave wants to ask, is artificial intelligence being adopted as a research tool?
Has artificial intelligence been adopted as a research tool? That's machine learning? Yes, that is literally what I use.
We need to move into the next speaker, but we have plenty of time for questions in the short break and also during the lunch. So Nabila will be looking forward to answer the remainder of your questions.
Please do come forward later.
Okay, so hello everybody. I'm Dr. Daniel Madison. I work in the Smith Group here at the Dementia Research Institute. And today I'm going to talk to you about how I use fruit flies to study Parkinson's. Yes, you heard right, fruit flies.
You get shaky wings.
Okay, so as Nabila beautifully just presented to you, she takes samples from Parkinson's individuals. She can study them to see not only what is happening to people with Parkinson's, but as we've already discussed, try and figure out as well what is causing Parkinson's. We can take that information and then try and model it in other systems to help us try and get into a deeper understanding of what is causing Parkinson's and what is then causing the development of Parkinson's. And so I work on fly models, but then you'll also hear talks later on from Harry and Matt that work on mice and human cells. So I know what you're thinking. Is our laboratory just full of flies flying around the room? Well, of course, that would be chaos. We don't do it like that. So we have many of these small plastic vials that we keep the flies inside with plenty of food. And when we want to look at the flies, we use a gas, carbon dioxide, to temporarily anesthetize the flies. And that allows us to look at them through the microscope and keep them still while we're trying to study them, so we don't have them just flying around all over the place. And as you can see here on our shelves, we have many, many of these vials of fruit flies. And each of these vials contains flies which has different genetic material, different DNA, just like us as individuals. And this allows us to see how differences in our DNA might be contributing towards causing or developing Parkinson's disease. So how on earth do we use fruit flies to study human brain disease, and why? So I'm hoping to show this to you in my talk. And the first reason that we use fruit flies is just for speed. They're really, really quick to use. So from 2 adult flies mating to the next generation of flies being produced takes just 10 days, which means we can really quickly manipulate the genetics of these flies and have the next test ready to go very quickly. And also, because healthy adult flies only live for about 3 months, it means that we can test the effects of aging, again, in a relatively short amount of time, which means that we get answers very quickly, which we can pass on to other researchers to help to understand what's going on in Parkinson's disease. They're, of course, quite inexpensive to keep. We just need those plastic vials and the food that they eat. That's basically all of the equipment we need, so it doesn't cost too much to do this research. And also, believe it or not, flies have quite similar genetic material to us as humans, which means that we can compare things that are happening in Parkinson's individuals to flies and hope to learn something from that. So first of all, I mean, this discussion has already come up, but just in order for you to understand this, I just want to take some time to explain exactly what a gene is. So if you consider your DNA, your genetic material, to be like an instruction manual with instructions for all the things your body needs to produce and maintain in order to function, you can consider genes to be like little subsections in the table of contents of this instruction manual. So small sets of instructions for single tasks or things that need to be produced. Unfortunately, what can sometimes happen to these genes is they can undergo mutations, which are basically like typos. So things that have gone wrong in the text of your genetic code, which means that the body is then unable to interpret them. It doesn't understand what it needs to do, and that's where things start to go wrong. And we think that, as we've already discussed, that there might be a genetic component to the contribution towards Parkinson's disease. So we think that there might be environmental, but also genetic factors that contribute towards the disease. And we've been discovering genes that we think contribute towards Parkinson's disease for over 25 years now. So the first one was discovered in 1997. We're discovering new genes all the time. And due to recent advances in technology, if you look just in the last 5 years or so, we've discovered over 100 genes which we think contribute towards Parkinson's disease. So really our understanding of the causes as well as the downstream consequences of Parkinson's are improving all of the time. But today I want to go back to 2004 to focus on one of the first identified genes which we think is involved in Parkinson's. And that's a gene called PINK1. But I should just point out that if you look at those first 10 genes that have been discovered, which we think are contributing towards Parkinson's disease, flies have 8 out of 10 of those same genes. So again, like I said before, we have more in common with flies than we might think. And this is why we can use the flies to study Parkinson's and have some confidence that things that we see in the fly, we might also see in humans too. So we can take that shared genetic material between flies and humans and also look at things that happen in Parkinson's that might also happen in flies. So things like movement disorder and the loss of brain cells. And so if we give our flies a mutation in the gene PINK1, like those that we see in some forms of Parkinson's, we can look to see how those flies perform in terms of their ability to move and whether their brain cells are becoming lost or not. So first of all, to look at movement, I'll just show you this short video to explain what we do. So this is an assay, a test built on the fly's instinctive response to try and escape from the vials that we're keeping them inside. And when we tap them down to the bottom of you can see on the left here, healthy flies, their first instinct is to climb straight back up the side of the tube and try and escape out of the top. But if we compare this to flies that have mutation in the PINK1 gene, like we see in some forms of Parkinson's, I don't know if you can see, there are, I promise, flies in the tube. They're all at the bottom of the vial and they can't climb up the side of the tube. And then if we look at the loss of brain cells, so we can label in the fly's brain dopamine cells, which are the ones that we think are being lost in Parkinson's disease. And again, if we compare the number of dopamine cells in a healthy fly compared to a fly which carries a mutation in the PINK1 gene, we can actually count these cells. And in a healthy fly, we typically see 12 dopamine cells on each side of the brain. And if we compare this to flies which have the mutation in the PINK1 gene, we typically see only about 9, indicating that they've lost some of these cells. So why exactly is this happening? So we've learned in the fly and also in other systems that what we think that PINK1 is involved in is maintaining a thing in your cells called mitochondria. So mitochondria are the things that give your cells energy. They allow the cells to function as they need to do. And I want you to consider mitochondria like the battery in an electric car. And I use this specific analogy because a battery in an electric car isn't actually one battery, it's multiple batteries. Multiple batteries making up modules and multiple modules all inside the car. And what that allows us to do is if one battery becomes broken, and we can replace that battery without the whole car breaking down. And indeed, if a whole module becomes dysfunctional, we can take out the module, put a new one inside, and the car still runs. And this is what we think that PINK1 is doing to our mitochondria. It's taking the damaged mitochondria out of the cell and replacing it with new healthy ones. So then if we think about what this is doing to our brain cells, you can consider a healthy brain cell to have a fully charged battery. And we think that in Parkinson's brain cells, because PINK1 isn't able to replace those damaged mitochondria, the cell essentially becomes like a drained battery and it doesn't have the energy to process as it needs to do. And I was mentioning to you today just one gene, PINK1. But we also have done research on many of these other genes which we think are contributing towards the progression of Parkinson's. And actually, if you look at all of these genes, all of them cause mitochondrial dysfunction in one way or another. So we think that things that we can learn from PINK1 might also be relevant to other forms of Parkinson's. This might be a therapy that we can use more broadly than just people that carry the PINK1 mutation. And so how might we go about improving the situation for people in this position? And what we've discovered is that if we give these fruit flies that have the mutation in the PINK1 gene extra copies of another gene, which also serves this function of replacing damaged mitochondria, then we can actually reverse the loss of dopamine cells in the brain. So here, if you compare again the healthy fly to the one carrying the PINK1 mutation that we see in Parkinson's, these flies have fewer cells than the healthy ones. If we give the flies that carry the PINK1 mutation extra copies of another gene called Parkin, we can reverse that cell loss, and these flies also have 12 dopamine cells in their brain. And we're looking to see if we can do this kind of therapy in other systems, like first of all testing in mice and human cells, and ultimately see if this could be a treatment that's effective for treating Parkinson's. So that's basically my talk. So I want to thank you for your time and happy to take any questions.
So they know I'm a chopsy one. I have questions. It may seem a daft question. It probably is a daft question. But we were at an event on Friday where they were talking about diet and the way that diet can affect different people differently.
Mm-hmm.
When you're feeding your flies, do they all get the same diet?
Yes, it's actually a very good question. So I will just repeat the question. So the question is about diet and whether the diet of the fly is controlled, because that could be something that is potentially having an influence on the development of Parkinson's. It's a very good question. So yes, we control our diet here in Cardiff because it's not something that we're specifically looking at ourselves. However, there are other labs where, for example, they might not do any genetic manipulation at all, but they do manipulations in diet to see what effect that might have on the progression of these symptoms. So yes, everything that we do here is on a controlled diet, but it is possible to manipulate, change components of the food, and then see what effect that might have as well.
Why did you choose to add extra parking rather than changing the PINK1?
Yeah, that's a good question. So the reason is a little bit complex. It's essentially, I'll go back a few slides. Just to this timeline, just to highlight the fact that not only is PINK1 a gene which we think contributes towards Parkinson's disease, Parkin is one of those genes too. And what the fly was actually instrumental in showing is that Parkin serves its function in the body after PINK1. So it's possible for Parkin to operate in the absence of PINK1, but not the other way around. So it's possible that you might be able to treat people with either a Parkin mutation or a PINK1 mutation with extra copies of Parkin, which is why that's kind of more of a tractable option than giving PINK1, because it will kind of be able to treat both of those patients rather than one.
I have a question from Colin, from the virtual attendees, that says that your talk was fascinating. And if you can link the different genes involved and labeling fruit flies to any specific symptom.
So just to repeat the question and try and understand it myself. So can we look at all of these genes as a collection that contribute towards Parkinson's disease and find a common symptom that links them?
I think the question might be, does PINK1 cause a certain type like tremor? Does LAMP2 cause cognitive?
As far as I understand it, so basically, can you pinpoint specific mutations that cause specific symptoms of Parkinson's? The way that I understand it is kind of that mutations in all of these genes ultimately cause Parkinson's disease, of which there is quite a tight definition clinically of what Parkinson's disease is. So I think it's actually quite hard to pick out which genes might be contributing towards which symptom, because at least several of these genes can cause Parkinson's disease on their own, not as a collection of mutations. So I don't really want to get too deep into this, but some of these genes that have been discovered in more recent years might not be causing Parkinson's disease on their own, but as a collection. So it's possible from looking at some of these more newly discovered genes that we might be able to pick apart genetic contributors to specific symptoms, but we're not quite there yet.
So there's another question from Elizabeth, which is essentially asking, how would you utilize this as a therapy? How would you give somebody more copies of Parkin? Would you use something like CRISPR?
Like CRISPR? I mean, CRISPR still at this stage is a long way away from being in Parkinson's disease patients, I would say. I think there's been some work, at least in mice, using essentially safe viruses, so viruses that don't have any pathogenicity, but you can include genetic material into those viruses and then use that as a mechanism for getting extra genes into the brain. Again, this is something that's quite experimental, and I don't think is being used in any human therapies at the moment. But possibly, again, somewhere down the line, we might be able to come up with a mechanism like this for doing gene therapy in people without using CRISPR.
Sorry, what is CRISPR?
So CRISPR is a kind of quite newly discovered way of editing people's DNA. So you can basically, if you imagine someone's DNA being written in a computer, you could go in and delete one letter of that code and change it with something else.
On a human basis or on a fly basis?
So at the moment, we can do it in flies, we can do it in human cells. Doing that in a living human is still quite controversial, because we're obviously very concerned about what kind of side effects might be caused as a consequence. So we need to do a lot more tests before we think that that's safe to do in people. But maybe sometime in the future, then that might be possible to do.
I recently had a DNA test of several potential relations. I found out that my half-brother, who I met after 60 years of search for him, has Parkinson's as well.
Mm-hmm.
Is DNA used at all doing research of Parkinson's?
Yes, quite a lot. So, I mean, our ability to build these models to study Parkinson's disease is built off understanding the DNA that people with Parkinson's have. So if we didn't have that genetic information, we wouldn't be able to build these models because we wouldn't know which genes to change. So genetics is a really strong part of our research, especially here in Cardiff. We do a huge amount of work looking at the ways that DNA might contribute towards the disease.
And can you also follow the relationships between the various?
Yes, absolutely. So it kind of varies gene to gene. So some genes might have a very strong hereditary factor, so they get passed down, and every generation that possesses that mutation in the gene might have the disease. Sometimes it's more complicated than that, so maybe we'll skip generations or just pop up from time to time.
It's just hit me and half brother.
Yes, yeah, absolutely.
So now we're going to have a little bit of time for a comfort break. So please, everyone, take a seat, and we'll start with the second part of our talks. Harry is going to tell you about his research using mouse models to study Parkinson's disease. Thank you.
Hello, everyone. I'm Harry. I'm a researcher here at the Dementia Research Institute, working in the Picano Kelly lab. Sorry, Dave. And we study the effects of Parkinson's using mice. So why do we need to use mice? Well, first of all, the law states that we can't use mice in experiments if the information we can get can be obtained from anywhere else in any other way. So computers can tell us which cells to focus on. Cells can tell us how Parkinson's affects them and how they work. And flies can show us how the Parkinson's cells work as a system. So what can mice tell us that computers, cells, and flies can't? So in people, brain cells are not alone. They work as part of a team, and these teams work together across the whole brain. And the brain controls our whole body. So when something goes wrong at any of these levels, it can affect how we think, how we feel, how we sleep, and how we move. And mice are an excellent way to look at these behaviours. So computer cells and flies tell us what we need to change in the mice to simulate Parkinson's. But can a cell do this? And can a fly do this? As you can see, the mice they're very smart, and they can learn to solve really complex problems as long as they get the right reward, in this case, seeds. So our mice slowly develop signs of Parkinson's as they get older, and this makes them great for studying how Parkinson's affects how we think, learn, and problem solve over time. So while there may be differences, many parts of the mouse brain work in the same way as ours do. They do similar jobs, and they can go wrong in a similar way. In our group, we're focusing on these non-motor signs, as they typically appear earliest in people with Parkinson's, such as problem sleeping, anxiousness, memory issues, and thinking less clearly. So I'd like to show you one of the ways that we can measure these things in mice. Okay. Here we have a puzzle box. It has two rooms that the mice can go into, one of which is brightly lit, and then the other one here is nice and dark and cosy for them. When we place a mouse into the light part of the box, they tend to want to run straight into the safe dark space and relax there. So to measure how well these mice can think, we can put little problems in the way of the dark area, and they need to solve these in order to get in there. In time, how long it takes them to solve these problems. So the first test is easy. Put them in the box. There's a little tunnel there they need to go through, and he's done it. So next, we can then block the tunnel with something like sawdust. This is sped up, because they're not really this fast. So he knows that he wants to get into the box. He's trying to work out what to do. Eventually, he'll work out that he has to dig his way through into there. We can then return the mouse to the box a bit later and we can see how well they're remembering what to do in this situation. This is in real time. Go straight in there, dig its way through. So we can then increase the complexity of the tasks and really put them through their paces. So here, the mouse needs to work out, needs to pull out a stopper from the tunnel in order to get behind it and get to a safe space. Takes a bit longer because it's a bit harder for the mouse to work out, but eventually works it out and goes in. And then again, when we repeat the test a bit later to see if they can go straight to the plug. This is the right key. Okay. Pulls the plug out and gets into a safe space. So we can look at the way that the Parkinson's mice solve these problems over time as they get older and how well they can remember the rules that they need to get in there. So right now we're measuring how they do in these tests, when the problems might begin to show in their lifespan. And once we do know, we can see how various Parkinson's treatments might improve their performance or maintain their ability to learn and remember. So using tests like these shows how behaviour can be changed by Parkinson's, and we can measure how thinking, feeling, sleeping, moving, how all of these things change over time. We try to detect the earliest changes we can in order to test early treatments, and this could then help to slow or even reverse signs of Parkinson's in the mice. And then in turn, this will inform us which treatments might be possible to move forward into the clinic then. There we go, that's the end of the talk. So any questions about the mice?
Question for me is the last point of the board is testing treatments which can slow reverse these changes. So how far advanced are you in terms of your thinking and your analyses in testing those treatments, and what are the treatments?
So there's lots of different models of the mice and lots of different treatments. Oh, sorry. So the question was, how advanced are we in testing these treatments, testing treatments for Parkinson in the mice?
And what are they?
And what are they? So yeah, there's lots of lots of avenues being explored by different groups. So at the moment, with this mouse model that we're talking about here, at the moment we're just seeing how the disease develops over time at the moment. Once we know that, we can then maybe test some treatments to see how it changes. So these might be drug treatments, or some groups might be looking at the deep brain stimulation, for example, or things like diet and exercise and these other interventions like that. So a whole world of possibilities.
How different are the mice? Does mouse X, who does the stopper test quickly, do other tests just as quickly, whereas mouse Y struggles with?
Much like, so the question was, how different are the mice to each other doing these tests? So if they're good at one test, do they then go on and be good at the other tests? And are there any differences, basically? And yes, much like people, there's a range of abilities in the mice. You get some slow ones that can't quite work it out, some that can do it straight away. So that's why we test groups of mice. You can see the whole range, and in general, we'll see a healthy mouse group as a whole will complete a task quicker. There might be individuals in that group that be slower, but in general, that group might perform it quicker than, say, mice with Parkinson's. But again, in that group of Parkinson's, there might be some which are really good at it.
Who are you actually giving them the Parkinson's with? Genetic?
Yeah, so with mice, it's relatively simple to create genetic models. So we can look at the genes that have been found in the past and insert them into the mouse DNA and see how they affect mice as they grow and develop.
So if you actually give the genetic But I thought they were sort of more like not causes, but make things more likely. If you make a genetic change to all of the mice to get Parkinson's, is it worth?
So the question was, if we make the genetic changes in the mice, do all of them get affected by Parkinson's? In general, we see that the majority of the ones that carry these changed genes will go on to develop some sort of signs, again, to different degrees, and it does depend on which gene you're looking at or set of genes. Yeah, there's quite a lot of variation, so we need to sort of have a broad group.
There's a question in the chat, Colin Young, I'll abbreviate it effectively. We'd like to know how similar brain structures are in mice to humans.
The question was, how similar are the brain structures between mice and humans? Mice and humans share a great number of cells which do the same job. They also have the same circuitry, similar circuitry in the brain, which work together to do similar jobs in the mice. So while they might be a lot smaller, it's far fewer cells, less complicated. Yeah, there's huge similarities between them. We can match the signs that we see in the mice with what we see in people.
You're looking at the brain structure. What about mitochondria? Are you following on from your colleagues who are looking at flies to see whether mice get the same effect on mitochondria in their cells?
So there's definitely research going on in that region. My group specifically aren't looking at mitochondria, but it's definitely possible to do that in mice. So we can take the cells from the mice and look at the mitochondria and see how they're changing.
That's fantastic. I think we can move to our last speaker of the day. And if you have a question,
so I'm the last speaker. I'm going to try to be short and concise because it's actually a bit late. So I'm Matt, and I'm a neurostem cell research associate in stem cell neurobiology in the lab of Cardiff University. And in the lab, we're using actually stem cells to try to understand how Parkinson's, what causes Parkinson's using human stem cell model. So as I said, I'm working with human stem cells. And as you all know, in the body, we have a lot of different cells which are highly specialized and to do only one thing. So the muscle cells will only be like muscle cells. The skin cells will only be a skin cells. On the other hand, stem cells, they have the ability to become any type of cells within our body. And we can take this actually great analogy with children when they're at school. They have no job yet, and they try to learn new things. So as parents, we can actually encourage them to go towards a specific path. If they want to go to become a lawyer, they need to go to law school. Farm school for farmer, mechanics, or surgeon needs to go to med school. Well, stem cells, they're actually exactly the same. We need to choose what they're going to become. So in our lab, with our stem cells, we can, for example, add drug A. And drug A is actually going to generate human brain cells. If we add drug B, that can become a muscle cells. And if we add drug C or D, can become a lung or kidney cells. So the very important take-home message here is stem cells have the ability to become any type of cells within our body. So, as you know, Parkinson's is defined by lots of cells within the brain, and the brain is composed, like Harry showed you, of a multitude of billions of brain cells that are all connected together and actually help us to think, to move, and to do everything we do every day. And when in a healthy individual, you have in the brain a region here composed of brown human brain cells that remain the same all along your life. There's no loss. On the other hand, when you have Parkinson's, this region, you can clearly see there is a very strong loss of these human brain cells, which subsequently will lead to all the motor symptoms that you're all aware of. So as Nabila previously told you, Parkinson's is a very, very complex disorder. And as also Daniel told you, Parkinson can be caused by a lot and multitude of different genes. So on the other hand, using compared to all the other models that have been actually presented to you using fly model or mouse model, by using stem cells, we can actually study you and your DNA and the way you're actually unique. So if we take the example again of Susan and Tom, they're developing this symptom very differently. One, like for Susan, she's going to start developing a symptom at 55, while Tom is starting at the age of 42. So what caused the differences in the appearance of the symptoms? So if we actually use stem cells, we can take a biopsy of your skin or blood, and if we use a very specific cocktail of drugs, we can actually create these stem cells that will have the ability to give rise to every type of tissue we want that we choose for them to be. So, as I told you, from these stem cells that we have here, we can add the drug A, and the drug A is going to create human brain cells. And once we have these human brain cells, we can actually study, like Daniel is doing, mitochondria. And mitochondria, just to remind, is like the powerhouse of the cells that gives all the energy to your neurons, to your human brain cells to properly function. And so usually in Parkinson, they are affected, therefore the production of energy is deficient. And in this particular example here, using the pink one gene that's been demonstrated to you by Daniel, when we look at healthy individual, you have in green the mitochondria. Here there is a zoom for both regions. And in healthy individual, you can see that all these mitochondria are very well connected, all connected with each other, creating a very nice network. On the other hand, with pink one, you can observe here that all these mitochondria are more like small blobs. They are not very well connected with each other. And we can, again, take the example of the battery. The battery of the car needs a multitude of different batteries to work well together. And when you have all the different batteries here, your car is going to work very well. On the other hand, if you're missing 1, 2, or 3, or 4 batteries, you're going to have a difficulty with your car at some point, which is the case in Parkinson. So just to conclude, and the very short take-home message is that with stem cells, here at Cardiff, we have the ability to generate human brain cells. And we can generate human brain cells. And because we can actually take cells from you as patients and also a healthy individual, we can study you as a unique individual, and we can actually identify and understand why you will actually develop Parkinson earlier or later on. And that was very short, but concise.
Is all the research that's been done, and not just you, but with your colleagues as well, is it being shared with other centres throughout the country or throughout the world?
Yeah, like all our work is very, especially at the DRI, everything is very collaborative. So we all work, like we always have like a number of collaborators in which we cannot do all the tests on our own. Sometimes we need to have other teams that actually help us understand what's actually happening within our model. So yeah, the data are usually shared. And also, as you may know, when we found something, we usually write publication to be spread across so that other researchers can actually read what we've done and not necessarily do the same things, trying to understand a bit more. Yeah.
It's the same question, basically. Do we have global research, global assistance, access to information, so on? Other
diseases seem to have priority, but of course they may not be so complicated. HIV and the recent viruses. They can find a solution for these quite quickly. But Parkinson's has been with us now over 100 years. Doesn't seem to be making much progress. I can understand when you look at the blueprints, my words for your diagrams, when you go into the structure of the cell, etc. I can understand why. It seems a pity to be not making much progress.
Yeah, so the question was, the remark was that we, compared to other pathology, there is less sometimes done in terms of Parkinson's study. And like especially for Parkinson, it's very extremely complex. As you can see, there is more than 100 genes that can actually cause Parkinson's. So you are actually probably most likely are different forms of as well between you of Parkinson's. So to find a common treatment, as Nabila showed you, is almost impossible. So that's why we need to study every single form of it and try to come up with some sort of treatment that can be used for a specific mutation, for a specific gene. It's very difficult for us to find like one single.
But there's thought that perhaps Parkinson's is one of those things that you have and keep quiet about it and carry on. Or people don't want to know too much about it and carry on. Whereas other diseases seem to have a pronouncement, you know, where people are aware of it.
Yeah.
Can I ask you, where do your stem cells come from, please?
Yeah, so most of our stem cells are coming from individuals that gave them to us. So we had some people in the clinic that can come and see the clinician, and we can, like, the clinician can actually request whether they will be happy for them to actually provide us with cells that we could actually use after. So they are human cells, they are your cells, so they contain your genetic material, your DNA, they are unique for you.
Thank you.
Can I ask about research aims and joining up research in different fields?
Sorry?
Joining up research in different fields. Well, one thing that strikes me when looking at Parkinson's is, and I've asked the question, what are the 2 or 3 things that are most beneficial in treating Parkinson's? And I've been told one is to exercise more. Second one is to watch your diet in terms of nutrition, best nutrition. And the third one then is the drugs. So how has that area been looked at in terms of joining up those remedies?
To find new drugs, you mean?
Not just the drugs, but the impact of exercise. And I think we discussed diet earlier. But
yeah. So in my field at all, like my cells are in a petri dish, and for me, they are staying there. I cannot actually assess exercise in my cells on my cells. So, but I know that there are other teams probably also working in Kubrick that are, I'm sure, looking at the impact of exercise on Parkinson, yeah, but not necessarily here.
Yeah. If you take a stem cell and you make a phase to it to try and make it into a brain cell,
yeah,
from a particular person, if you do it once and then you do it again, are the 2 resulting cells the same, or are there sometimes variations?
Okay. Yeah. So when we do it, that's what I was doing. When we're having the stem cells, are our results actually reproducible? Can we get one result more than once? So that's something we are obviously looking at as well, and that's why we have very detailed protocol in which what we're doing is very standardized, and hopefully when we get our human brain cells, they are all the same across different batches. That's why we need to be very careful about that, so to not introduce any variability. So that's.
So we can thank Matt for the wonderful talk, and we'll have more time for questions during the lunch. Our chat is open also for the virtual attendees to send us more questions, so they have any, and we'll keep it open for a few extra minutes. So now I just want to finish this session. Thank you all for listening, both here in person and virtually. Here there is some ways to contact us. If you have any other questions about our research, or you can know more about our Dementia Research Institute. I'm also sharing you this website that can point you towards how to join, for example, the Welsh Research Interest Group, and also this website with more information and easy to understand about Parkinson's disease research, and some other websites to join for trials or any research projects. This is going to be recorded, so you can browse these websites later. And finally, also saying that in July next year, the World Parkinson Congress is taking place in Barcelona, and this one is open for all researchers, people with Parkinson, carers, etc. And finally, just wanted to close this session with a poem participating in the campaign poems for World Parkinson's Day 2022. This poem is by Jeff Flores, that I think is attending online to the event.
Hi, my name is Jeff Lawrence. I'm a volunteer with Parkinson's UK. I've had Parkinson's now for 7 years, and I've taken to writing poetry, hence I've been asked to read a poem on this video today. I like to know when people write poems, what inspired them to write the poem, so I'll give you a little bit of introduction. This poem was written just after my diagnosis. In fact, it was the first poem I ever wrote. And it's dedicated to my wife, Anne, who, in her own words, nearly threw up on the consultant's desk when I was diagnosed with Parkinson. By nature, Anne is a worrier, and she just can't switch off. But she's been very supportive to me over the years, as the years have passed and my symptoms have got a little bit worse. One morning, we were just lying in bed. I was awake early, as I normally am, earlier than Anne, and I was just watching her sleeping on the pillow. And the pillow beside me, and I just was inspired to write this poem. It's called I Wonder What She's Thinking. So I hope you enjoy it. Here we go. I wonder what she's thinking as we lay here in our bed. The thoughts are always swirling round and round inside her head. We raised our daughter Ian, taught her till she flew the nest. Nursed our parents when they were frail and old. We did our very best. Our grandchildren then came along. We more than played our part. Read books and played games while they pulled strings on our heart. But now they're both in school, and whilst it was great fun, it's our time to relax and celebrate a job well done. But Parkinson's arrived and took control of our hands and stuck his big old spoke in our retirement plans. Why is life so unfair? Anne often says to me. Why ask why now, she asks, and I tend to agree. But we talk it all through, now it's just you and me. We can do this together, we're strong. But I know as she fears it'll all end in tears, there's so much that could go wrong. So hold her and say, let's live for today. The future, well, that's for the birds. I squeeze her hand tight, say, come on, where's the fight? Your thoughts for today, they're just words. I give her cuddle, her head's in the middle, but my thoughts are all very clear. Her is my rock, there'll be no future shock. I know we have nothing to fear. Whatever our fate, we'll be sure to create a bond between us strong as steel. We'll face it together, any storms we will weather. No parking can spoil how I feel. So out here in my heart, nothing drives us apart. I'm sure that we'll both discover. Love you more every day, so just wanted to say, you're my rock, my best friend, and my lover. There you go.